Okay, welcome to Let's Get to the Marks. Time for an A-level biology lesson on the structure of a chloroplast. So let's go. Here we have the outer membrane, followed by an inner membrane, sometimes called double membrane or an envelope. And that's why it's found in eukaryotic cells. You wouldn't find that in a prokaryote because you don't really find double membrane bound organelles in prokaryotes. Inside we have a gel-like substance, a kind of jelly. We call this the stroma. It's made of, well, it contains enzymes and organic acids. And you'll also get the odd starch grain floating about there, which is where some unused glucose has been joined together to make the polymer amylose or amylopectin for storage. It's also the site of the light-independent reaction, or the Calvin cycle. You know, the bit with ribulose, biphosphate, ribisco, and carbon dioxide. That business is going on there. Okay, here's an unused starch grain. Then we have the thylakoids. These are membrane-bound sacs filled with a fluid, but also they carry photosystems on their membrane. That's proteins bound to a pigment like chlorophyll A or B, and that's where the light is absorbed and the light-dependent reaction occurs. Now, they don't occur in ones, these thylakoids. They occur in stacks, like putting 10-pence pieces or £1 coins on top of each other, and we call these granum. Granum for singular, plural grana, which is a stack of thylakoids. And then these stacks are joined to another stack via an intergranal lamellae, or lamella, which is the structure that joins two of these stacks of granum together. So intergranal lamellae link the grana together. And finally, not really on the syllabus, but something that you should be aware of, that chloroplasts, a bit like mitochondria, but chloroplasts contain their own DNA, their own genome. They have circular DNA. Some chloroplasts in algae have plasmids, others have circular DNA, like plant chloroplasts contain circular DNA. And yeah, if you want to look more into that, you can look about the theory of endosymbiosis, how once upon a time chloroplasts were organisms before they came into other cells. Okay, let's go through a question on the chloroplast. So 22A, the image is a transmission electron micrograph, TEM, of a chloroplast. So this is the structure of what the diagram we've just been looking at, basically. Complete the table using the letters from the TEM, which identifies the structure or function. Each letter may be used once, more than once, or not at all. So the structure or function, you've got all of those down here, and all you've got to do is pop a letter in each of these. So pause the video, have a go. All right, let's go through the answers. Chloroplast envelope. Which letter points to the chloroplast envelope? Well, that is the double membrane, so that's going to be F. Where do we find the light-independent reactions? The site of the light-independent reactions. So that's the Calvin cycle, for those of you that know it like that. And that is found in the letter E. That's the stroma. Which one of these letters points to the intergranal lamellae? So we're looking at a structure that joins two stacks of thylakoids together. You're looking at D. So you've got the granum on either side, joined together by the lighter colored gray kind of lines. And that is the intergranal lamellae. And then which structure above contains starch grains and lipid droplets? Didn't mention lipid droplets when I went through the diagram, but the starch grains should be the giveaway. Which area contains the starch grains? That'll be E again. So notice it did say we don't have to use all of these letters. So G here was not used. Of course, G is pointing to the granum, which is a much more highly dense structure than the intergranal lamellae. So it's come out in a darker color. Awesome. All right.
You're still here? It's over.